Hello everyone, welcome to Your Soil, Your Data. I'm Dr. Deborah Long and I'm Programme Director at the Grow Observatory. I'm an ecologist and I've spent my career working with charities in Scotland focused on conserving natural and cultural heritage with a particular focus on native plants and wildlife conservation. In my garden, I grow native and garden flowers and food plants for me and the local wildlife. Thank you for coming along. Some of you joining us today have taken part in our activities last year and in the online course that's happening now, Living Soils, Growing Food. For others, this will be your first encounter with the Grow Observatory, so welcome. Grow is a citizens observatory which is taking citizen science to the next level. Citizen science isn't new. If you've taken part in the Garden Bird Watch or the Big Butterfly Count, then you've done some citizen science. Grow has a big ambition, though, to build a community of thousands of citizen scientists to address the urgent challenges of soil degradation and soil management, as well as resilience to climate change and sustainable local food production. We're aiming to benefit the practice of individual growers, as well as help answer some of the major challenges for science. And you can be a part of Grow's two main citizen science missions happening this year. One is our Changing Climate mission, which takes place in 9 Grow places in Europe. You can find out more about Grow places, where they are and how to get involved by looking on the Grow website. And our Living Soils mission. You can take part in this mission wherever you are. And this includes our Grow experiment. We'll tell you more about that later on. You can also take part in discussions and activities on the Grow website and the 4 online courses over the course of this year. So on the webinar today, I'm joined by 3 members of the Grow team. Please know our presenters are all over the world, so you might experience a slight delay between speakers. However, let's give it a go. Let's start with Alice.
Hello, I'm Alice Ambler. I'm an early career researcher with a lot of interests. I live in Scotland and I'm a growing beginner. I'm especially interested in using organic and regenerative practices to improve my soil for the future. I'm also interested in water-related environmental hazards, some of which may impact on growing, such as floods or droughts. Over to you, Pavlos.
Hello, I'm Pavlos Georgiadis. I'm an ethnobotanist, biodiversity researcher, and organic olive grower. My passion for growing and good food and its communities has seen me travel in several countries across the world, and I'm now based in Greece. I'm the Grow Community Manager, working with a network of Grow Community Champions across 9 European countries to deliver Grow's Changing Climate mission. Over to you, Victoria.
Now based at the Permaculture Association in Britain. I'm also busy writing up my PhD on how soil biodiversity responds to land use change. I'm particularly interested in the ecology of earthworms and insects, and I really believe that science is for everyone, and I'm keen to get as many people involved in doing experiments in their own growing places. Back to you, Deborah.
Thanks, everyone. In this webinar today, we'll talk about some of the key issues to think about when you're experimenting in your own growing space or conducting citizen science experiments. We'll discuss key issues around soils and growing that can help you in your growing and that also relate to global challenges based on what you said you were interested in during our online course. We'll tell you about some of the regenerative growing practices that we use, and we'll share some details about the Great Grow experiment and answer some of your questions about it. We also have about 25 minutes in this webinar to hear and answer your questions, so do please send them in. You can ask questions directly in the comments section of this live broadcast, on the Grow Facebook page, or via the Grow Twitter account @Grow Observatory using the hashtag #GrowWebinar. We've also collected some of your questions from the Living Soils online course. And then we'll look at what's next for the Grow Observatory and how you can take part. So let's get going. Over to you, Alice.
Welcome to everyone joining us today. Whether you've joined the ongoing online course or are simply interested in this topic and getting more involved, we hope you'll enjoy it. We'll start by talking a little bit about citizen science. Citizen science is the coming together of citizens to do scientific research. In the case of Grow, it allows large amounts of important data to be collected by people all over the world. In the case of an experiment, it can allow replication en masse, giving us robust scientific data. Grow has now run 3 online courses to train growers to become citizen scientists. This webinar is part of the Grow's current course, Living Soils, Growing Food. Through this course, we want to bring people together to do a citizen science experiment on growing a specific polyculture. Together, let's see if it works. Although the Citizen Observatories project, including Grow, are focused in Europe, we are pleased to welcome people from all around the world who wish to contribute to citizen science and learn more about their own food growing and soils. This map shows the locations of participants in our current online course. During the course, we've discussed the challenges and opportunities for global food and farming systems. We have focused on food growing, particularly on regenerative practices and other techniques that you can try in your own plots. And as part of the course, we've introduced ideas about how you can experiment with different types of growing, like comparing monocultures with polycultures. This is all in preparation for our upcoming citizen science experiment that we'll tell you more about later on. Evidence of the benefits of regenerative practices is increasing, and regenerative practices are becoming more and more widespread. However, when it comes to published papers and reports, the evidence often gained from these practices is often gained at a large scale rather than a small scale. This may seem unimportant if the practices are widely implemented anyway. If they work on a large scale, why would they not work on a small scale? Well, it isn't a given that the results will translate to a smaller scale, and there may be benefits or limitations only experienced at a small scale that we are unaware of. It can lead to questions such as, do you need a field full of legumes for them to provide any benefit in terms of nitrogen fixation? How many legumes do you need to provide benefit per, say, meter square? And what are the different benefits and limitations? This gap may also affect policies and funding support for campaigns promoting these practices. Over to you, Victoria, to tell us more about setting up your own experiment.
Thanks, Alice. So the first step in setting up an experiment is having a question to answer. Our question drives the whole process of how you set up your experiment, what you measure and how you analyse the results. Start with what you're interested in or what you'd like to find out, and do some research on the topic to find out what's already known. Maybe search on the internet, read some books, or ask others what their experience has been. Then think about what you can realistically do with the time and resources you have. The next step is to refine your question to one that's clear and answerable. Once you have your question or hypothesis, it's time to design an experiment. Experiments can be used to test a question by making manipulations or treatments and compare it with an area which has not been changed. We call this the control. Control plot gives you something to compare your manipulated plot with, meaning that we can be more confident that there is a cause and effect relationship between treatment and results. The control also helps rule out alternative explanations due to other variables and makes it easy to detect differences by reducing the variability between plots. But how do we know if the difference or not between the control and the treatment plot is real and not just due to chance? One way to do this is to repeat the experiment many times and compare the results. We also need to think about what we want to measure and how. For example, if we're interested in yield, do we want to measure weight, quantity, quality, or combination of these? We also need to think about measuring things that I'm not directly interested in, but may influence the results, such as soil and weather conditions. It's a good idea to think about what the results would look like and how we could display and understand them before we start. This allows us to plan what we need to measure during the experiment, because if you think of something else, it may not be possible to go back and do it. On the online course, we've had some really great ideas for questions and how to answer them using experiments in your growing places. We will discuss some of these now. So Alison asks, My question would involve how to stop the local fauna eating my food. How would they go about answering that, Alice?
Thanks, Victoria. This is a good example of a question that can be refined a little more into something that can be tested with an experiment. Firstly, we need to think about what local fauna. Are we talking about slugs or mice or deer, for example? Different methods would be used to control these. The next step is to find out about existing methods to prevent them from getting to your crops. Read up online in your or in your library and talk with other growers to see what they do. Consider which approaches are most likely to work for you. Think in terms of practicalities like cost, time, and how easy it is to do. Think of your ethics and the legalities. Do you want to kill the animals or just deter them? And also consider what is most likely to succeed on your particular site. Once you've narrowed it down to a few possibilities, you can set up an experiment to give them each a go and find out which is the easiest and most effective for you. I hope that helps you, Alison, and anyone else tackling the same problem, and have fun finding out. We have another research question suggestion from Cecily, who wants to try out 2 different composting methods, fermented bokashi and normal hot compost from garden waste, on 3 different crops to see if there are differences in growth, yield, disease, and importantly, slugs. Victoria, what are your thoughts on this?
So comparing 2 types of compost would make a really good experiment. Although Cecily could also consider having control with no compost at all to compare them with. They also need to think about other influences, keeping other influences the same as much as possible, such as using the same types of crops or the same number of plants. Cecily wants to measure a lot of different things, including some quite broad topics such as disease. It might be worth thinking which of these are of most interest and just concentrate on them. Whichever is chosen, they need to think about how they will measure it in a practical way. For example, if they're interested in slug damage, would they measure the number of seedlings lost or percentage leaf cover damage? They could also consider other things that might influence the results, such as weather or soil moisture. I hope that helps you, Cecily, and others planning this sort of experiment. Good luck with your learning. We have another good research question from Simon from Dursley in the UK, who wants to test the nitrogen contribution to the soil of various members of the pea family, such as runner beans, French beans, peas and clover. Pavlos, what do you think about this one?
Thanks, Victoria. I like this idea. It has a clear setup. We get a sense that the 4 legumes will be grown and then soil samples from each taken and compared before planting and later on to assess nitrogen content. Again, it would be also useful to add a control here, such as bare soil or possibly a non-legume crop that would help us to understand the overall contribution of the legumes to the soil and also to separate changes as a result of the legumes from any other background changes that might impact nitrogen levels. It's going to be important to find out a bit more about the expected differences in soil nitrogen and to ensure that the test we are using is able to discriminate at the right level. If the differences are going to be quite small, for example, it might be necessary to send samples to a laboratory for testing. The final key area to think about here is the time period. How long will it be before we are likely to see measurable differences? Do we need to wait until the crops have died down and released captured nitrogen from the roots? Will it take several years to see measurable differences? If you can't find this out from what is already known, then this could be an interesting question to start with. So, Simon, I hope that gives you some help with how to approach this. We have another experiment design from Alan, who runs a small eco-farm in Ohio, which I think is a great example. Alan is proposing to compare 3 soil preparation methods: conventional digging, double digging, and no dig with mulching, and to look at effects on soil organic matter, water retention, and nutrients. Victoria, perhaps you could tell us more about this?
Yes, so Alan's got a really nice experimental design set up. They've got a clear question to answer, and their method demonstrates important aspects of how to well plan an experiment. Firstly, they have a control plot which doesn't use any soil improvements at all to compare them against. They've got a clear plan of what they want to measure, and they thought about how to measure that. And they used some great use of technology with Wi-Fi connected sensors and laboratory tests to measure soil properties and weather before and after the growing season. This also helps minimise some of the variability by considering these other influences. So studying over several seasons is also a good thing to do because it is a form of replication, and also because sometimes soil improvements can take more than a year for benefits to be visible. Even better would be that if they could do replication with multiple plots of each soil improvement method, but this might not be possible due to time and space restrictions. Actually, most scientific experiments are a trade-off between the number of replicates and how much time, space, and other resources are available. This is where citizen science is brilliant, because we can replicate an experiment many more times than is usually possible by having lots of people try it out in their own growing space. So back to Deborah.
So we have more on the Grow experiment itself coming up, but first we'd like to share some of the regenerative growing practices that we use in our own growing spaces. Please remember to keep your questions coming in via the comments section of this live broadcast on the Grow Facebook page or on Twitter at Grow Observatory using the hashtag GrowWebinar. So first up, Pavlos, could you tell us how you use regenerative practices in your own growing?
Thanks, Deborah. It's always a pleasure to share what we are doing in that corner of Europe here in Greece. I get to practice several regenerative practices in my family's farm inside the ancient olive grove of Makri on the northeastern coast of Greece. Some of the trees in this grove are more than 1,500 years old and are situated in one of the 4 northernmost distributions of olives in the world. For this reason, it's important to continue their cultivation because their ancient genotypes are adapted to frost and drought and make up important knowledge for fighting climate change and ensuring olive growing in the future. My family has been cultivating these trees for 4 generations. We follow principles of organic farming and agroecology. The grove is not the typical industrial farming system where you see trees growing in rows, but rather an olive forest which we treat as a living ecosystem. We are working towards turning this olive grove into an adapted agroforestry system for the dry Mediterranean climate. For this reason, we continuously feed the topsoil, enhancing organic matter with mulch and compost, but also allowing a large biomass of understory vegetation to develop. When we cut these grasses, we let them cover the soil to protect it from high temperatures, especially during the hot summer months in Greece. And this allows us to grow with very little water, but also allows a large diversity of wild herbs and plants to grow inside the olive grove, supporting biodiversity and offering food and habitat for useful pollinators. We also need to consider space as a growing strategy. As with traditional olive groves with old large trees like ours, it is important to keep a generous distance between them to avoid competition of the root system for nutrients and also allow for better aeration between the tree branches, reducing the risk of pests spreading from tree to tree and eliminating the need for pesticides. Alice, what is your growing practices and tips?
As I mentioned earlier, I'm quite new to growing, but one of the things I'm excited about is planting legumes. Most legumes have root nodules that contain a bacteria called rhizobia. Rhizobia fixes atmospheric nitrogen and turns it into soil nitrogen. Legumes actually contribute approximately half of the biologically fixed nitrogen to the world's soil. Me and a friend grew some runner beans last year without really thinking about the benefits to the soil. One thing that struck us, though, was how abundant they were. This year, I'm going to plant legumes again, but this time I'm going to do it while thinking about their nitrogen-fixing potential. I have a few small plots to play with at the moment, so I'm going to try a few different methods. On one plot, I'm going to try using legumes as part of a crop rotation, while on another, I'm going to try pair them with squash, in the hope that the legumes will provide the squash with extra nitrogen. I want to use sugar snap peas and broad beans. I also want to use clover as a cover crop to add nitrogen and cut down the need for weeding. I think that it's important to realise how many different types of nitrogen-fixing legumes there actually are and how many different ways they can be used. They are most often used in intercropping, crop rotations, and as cover crops. They include many edible crops, such as beans, lentils, and many forage crops, such as clover. Victoria, what are your interests and tips?
So I'm interested in finding ways to encourage pollinators such as hoverflies, bees and butterflies. That's partly because I just really love insects and I love watching them in the garden, but also because many crops are reliant on insects to pollinate them. And so if there are fewer insects, I get lower yields or I have to bother pollinating things by hand. Providing food for pollinators in the form of flowers with lots of pollen and nectar is a key way to encourage them. Grow them flowers in sunny spots, as most pollinators prefer warmth. Avoid insecticides, which will kill them, and also avoid double-flowered varieties of plants, as they don't tend to have much pollen and nectar food. A succession of flowers providing a consistent food course is better than a mass in one go. Some of my favourite flowers for pollinators are Verbena bonaneris, Linnea purpurea, and plants from the carrot family such as Amy majus, or just leaving some parsnips or fennel to go to seed. Don't forget that pollinators are the adult insect and actually spend most of their life as larvae. You can help larvae by providing hollow stems for solitary bees to nest in, growing caterpillar food plants such as nettles and grasses, and tolerating some aphids on your plants which are food for hoverfly larvae. You could even try and breed aquatic hoverfly larvae by leaving out a bucket of dead leaves and water, although I don't recommend this if you live in an area with mosquitoes which can carry disease. Back to you, Deborah.
Thanks all. That was really interesting. And while you've been talking, we've had some great questions coming in, so do keep them coming in. We're going to start with one about string beans. And someone in Ashmead is given a question which says, what fertilizer treatment is recommended for string beans? Alice, can you give us some thoughts on that?
Hi, thanks. What a great question. Well, last year I got an amazing crop of actually runner beans just from using compost. Because most string beans actually create their own nitrogen, they don't actually need much fertilizing. They are in themselves an excellent fertilizer as they convert atmospheric nitrogen into soil nitrogen, ready to be taken up by plants. Try digging in a layer of compost into your bean patch and they should have some high yielding, and you should get some high yielding and fun and healthy bean plants. I hope that answers your question. Back to you, Deborah.
Thank you very much, Alice. We have another question here about how to measure nutrient uptake of plants and the general requirements of plants. Pavlos, could you give us some thoughts on that?
Yeah, sure. Thank you. Although there is no straightforward answer on how to measure the in plants, this question cannot have a straight answer. For plants growing in real gardening conditions, soil type and structure, which may vary even at the same plot, will play a role, as well as the presence of mycorrhizal fungi on the plant's roots, which will increase the absorption rate of nutrients. One way to test what works with your plot would be to experiment with plants that are inoculated with mycorrhizal fungi and some that are not. The difference in growth will offer some qualitative indication, but you will need to have access to laboratory equipment in order to have an accurate quantitative result. I guess this is also a matter of observation for growers out in the field. They are used in observing environmental conditions and changes in soil conditions in order to monitor the plant growth and hence make their own conclusions on nutrient uptake.
Okay, thanks, Pavlos. Victoria, we've had a question come in from Rod and Gail Crawford via YouTube, and they're interested in comparing dig and no dig approaches to see if it could improve their yields. What are your thoughts on that one?
Yeah, so this is a sort of growing things that can make quite a good experiment. So there's been quite a lot of research on sort of farm scales with different digging intelligence, but not so much on small scale. The main things you need to think about is your site where you're going to do the experiment and setting up your different treatment plots. So you could have one for the dig and no dig, and then you'd want to excuse me, make sure you grow the same sort of plants and use the same sort of fertilizers if you do that in each pot, so you can have a clear comparison that the actual method's making the difference and not some outside things. So it's useful to measure the things like soil moisture, texture, and nutrients if you can, and maybe the weather conditions as a sort of one of these additional variables that we need to consider. I'm happy to discuss more about this. It's just the sort of thing we want to get people into. So if you want to join the Grow discussion forums, I'm happy to chat more about sort of how you go about setting up an experiment like that. Okay, any more questions?
Okay, thanks, Victoria. We do have some more questions coming online. Pavlos, we've had quite a wide-ranging question here from Rihanna via Twitter, who's asking about the consumption of meat and dairy. If a massive decrease in our global consumption of meat and dairy is needed, how feasible do you think it would be to use a wholly vegetation-based system to feed our soils? Or would we still need animal manures to grow enough food? What do you think about that one?
Well, I think this is one of the most burning issues that humanity faces today, and surely there is not an easy answer. But I will attempt to give one, and I hope this will be satisfactory for Hernon. I think that it's important to consider here. What is important to consider here is scale. The FAO recognizes that the future of food growing is with small-scale farmers. Small-scale farming has, or at least should have, the ability to keep the balance between increased plant diversity with the presence of fewer animals. Those can enrich the soil with their manure, but also provide food in smaller, more sustainable quantities, but of higher quality. New technologies and innovations, including Grow's approach, can offer such tools to small-scale farmers, and hence they promise to play a major role into the future of food. But of course, this requires like a massive response from so many stakeholders active into the food system. But with the increasing awareness and new interest on food growing and the land that we are observing today, this is the great hope that we will be able to sustain food production in a way that is sustainable, it is ethical, and it's of course nutritious for us without ruining the planet.
Okay, many thanks, Pavlos. Excellent attempt at answering that massive question. We're going to move on to the experiment now, because we've got lots of questions coming in about those. So Victoria, can you tell us more about the Great Grow experiment?
Okay, thanks, Deborah. So polycultures are a really promising way of growing things. They offer the potential to provide food whilst also having environmental benefits. And scientific studies have shown that polycultures can reduce crop losses from pests and diseases, produce more food in a smaller space, and enhance ecosystems, biodiversity, soil and water conservation, and carbon storage. But most of these outcomes are not guaranteed, but depend a lot on what crops you're growing and where and how. And most of the research into polycultures has been done at the scale of large farms, where they use a lot of mechanized things to sow and harvest crops. There's actually a lot less scientific research done on the scales where most people grow, such as gardens, allotments, and smallholders. So lots of people do use polyculture. So many growers have experimented with companion planting, or they grow their crops quite mixed together. So there's loads of knowledge out there and experience. And it would be really brilliant if all of us who grow together at the smaller scale can join together to share this knowledge and investigate our practices. And we can do this through citizen science. So we want to address this gap in scientific knowledge with an experiment that you can take part in. The Grow experiment will compare 3 popular crops, so climbing beans, spinach and radish, grown together in a polyculture, compared with the same 3 crops grown separately. We are going to compare the yield quality and quantity from each plot, and there will also be opportunities to test soil nutrients at the start and end of the experiment. So if you join us, we can find more answers about your own growing space but also contribute to a wider understanding of polycultures at small scale growing. So you can sign up on the Grow website, and there's a link on the slide coming up. To join, you will need a growing space of about 5 square meters, which includes gaps between the plots. If you don't have access to this space or enough space, you could see if there's anyone who could team up with you, who do have enough space to try it together. Now, I know some people who've borrowed a bit of their neighbour's garden in exchange for a share of the harvest. Or perhaps there's a community garden in your area which would be interested in participating. You also need seeds of 3 different varieties. Phaseus vulgaris green beans, cultivar cobra, spinach, Spinacia oleracea, cultivar matador, and radish Raphanus saphaticus, the cherry bell cultivar. So these particular varieties have been chosen so that our experiments can be comparable with each other, and because these are really widely available ones. But if you can't get them in your country, you could use a similar variety, and we have some guidance on how to select the best one for that. And you could always ask us in the experiment forum on the Grow website if you're not sure. So quite a few people have said that these sort of crops won't grow for well in their area, but it might still be worth giving it a go. So it might be that they grow better in a polyculture than in a normal monoculture. And that would be really interesting to find out. We're also keen to help plan for future experiments with crops that are suitable in other people's locations. So if you have any ideas for that, please let us know through the forum. And even if you cannot do the experiment at this time, you're most welcome to follow it along as an observer, join in the discussion, and learn how to make your own experiments. So all of our experimenters on the Grow experiment will be given full instructions on how to set up the plot, lay out the seeds, and take the measurements needed. We're really keen that this isn't just about growing, giving scientists data. We want you to learn how to experiment in your own growing spaces, understand your results, and improve the way you grow. So over the summer, we'll be offering full and ongoing support for experimenters and observers to help you become a citizen science researcher. And in the autumn, we'll be exploring the results individually and collectively on our free final online course of the year in the autumn. So back to Deborah.
Thanks, Victoria. You've given us a really good introduction to the Great Grow experiment and how to join in. And we've had lots of questions about these. So let's make a start with some of these. And I'm going to come to you first, Victoria, with a question that I think a lot of people are asking. Jacqueline Wheeler from Surrey is wanting to know why climbing beans, radish and spinach have been chosen to, as opposed to, say, sweet corn, squash and beans, which is the traditional Three Sisters method. Can you tell us about that?
Yeah, so we've had lots of people ask this question rather than using the classic Three Sisters. And it's in part because the Permaculture Association did a small trial run using those 3 plants a couple of years back, but actually found that I think only one of the participants had any sweet corn from the crop, which obviously wasn't a really good outcome. So we tried to balance between 3 varieties which are widely available, people like to eat, and they also complement themselves in how much space they need and what soil they like. Any more questions?
Yes, we've got lots more questions. Another one for you, Victoria, and then I'm going to move over to Alice, I think. Another very common question, this time from Jana Holmstedt, not sure where Jana's from, but she's asking, does the polyculture experiment need to be 1 by 5 meters, or could it be laid out in an L shape?
Yeah, so because we're trying to keep everyone's experiment the same so we can compare them, it does need to be in the same amount of space and with the same amount of space between each plot, but the actual shape of the plots and the layout of them can be dependent on your growing space. So long as it's 1 by 5 meters area, it can be in any shape you need. So I hope that answers the question.
Thank you, Victoria. Thank you very much. And now we've got a question about slugs. So we have a question again from Ash Mead. Is there any possibility of including slug counting in the project? Alice, what do you think about that?
Hi, not at the moment in this experiment, but you could do your own slug counting during the experiment. It could be really interesting to see if there's a difference in slug numbers between the monocultures and the polycultures. And when counting slugs in your monocultures, you could see which crop attracts the most. Any more questions, Deborah?
Well, actually, I wanted to ask Victoria about that one, because Victoria, you mentioned earthworms earlier on. What about slugs?
I actually quite like slugs, except maybe when they're eating all my plants. But yes, we've had loads of people interested in counting slugs. So although that isn't something we thought about doing this year, it's certainly something maybe we're thinking about putting together some sort of measurement for in future experiments. It would be interesting to see sort of how the numbers and damage differs between different monocrops and polycultures. But remember, not all slugs are bad. I mean, some of them eat plants, but actually quite a lot of them are just only eat dead material, and they're important in decomposing your compost and things. So try and give them a bit of love.
So love your slugs. We've had a question about measuring soil moisture. Now, this is a different experiment that we think that we're running, and I think that's why Alan Roberts via YouTube is asking. He has a question about soil moisture sensors, and should he place the sensor in the row or outside the row, and how does he account for irrigation inputs? So I'm going to ask Pavlos to answer that, and then I've got a linked question about irrigating a polyculture bed that maybe I'll ask Victoria. So Pavlos first.
Thanks, Deborah. Thanks, Alan. So I think it depends on the type of experiment you want to do. If you want to measure moisture at any given plot, at any given site in your plot, you might as well choose to place it where you want. If you want to measure how efficient your irrigation is, then maybe it makes sense to place the sensor in the irrigated area. However, in what Grow tries to do, and that is validating satellite data, we try to aim for representation of the entire plot. If you want to validate your own sense data, your own measurements with what the satellites are offering, then the sensor needs to measure, to make a measurement that is representative of a wider area, let's say about 25-30 meters. And indeed, we are actually running now, we are launching now a mission in 9 grow places across Europe, where thousands of growers, citizens, will be using low-cost sensors to do exactly that. By the way, if you live in one of those areas, you should definitely contact us. And the names of the grow places are available on the growobservatory.org website.
Okay, thanks, Pavlos. Great plug there. Victoria, what about the additional bit of Alan's question, which is what's the recommended way of irrigating a polyculture bed? Drip, overhead, or other? Have you got any thoughts about that?
So I've not actually tried any irrigation in my own space, but I'm reliably informed by my colleagues that the best way to do it is with under soil type permeable pipes rather than an overhead one, because that helps keep the water actually in the soil where it's needed by the roots. And if you put it just straight onto the surface, it tends to evaporate. So that's probably, well, I'll take their advice that the in-soil one is the best way to go.
Okay, thank you very much, Victoria. We've had a couple of questions about radishes, and I'm going to start with a radish question that I can answer, and then I'm going to ask the next one. Caroline Osborne on Facebook is asking for good radish recipes. It's one of those crops that sometimes you get a good few too many. And there are some good recipes in the River Cottage website, if you look there. I found some to do with radish raita, which includes radish, goat's cheese, yogurt, and mint, which sounds delicious. Radish salsa, which is radish, mint, and spring onion, or even a radish and lettuce soup, which again sounds very interesting. So have a look there and see if there are some things you can do with your glut of radishes, perhaps. A second question about radish pods and the seeds in pods. Now, I'm not sure who will be able to answer this one, so I may well just ask one of our presenters just to have a bash. Radish pods are nice in a salad, and it's worth leaving a couple off of the plants. Frank on Facebook is asking if he could dry out the pods and use the seeds for next year's crop. He's wondering whether it would work as well as it does with runner beans. So has anyone got any answers for that? Alice, have you tried anything like that before?
No, I actually haven't. It sounds like a good idea and it sounds like it should work, but I'm not actually sure. Does anybody else have any ideas?
Victoria?
Yeah, I can't see why it wouldn't be worth giving it a go. The only thing to remember is sometimes if you're growing a particular radish variety, because they're cross-pollinating via insects, you might not get exactly the same type of radish come out of your seeds, but I expect it to still be, so it might be a bit of a gamble whether it would be nice or not, but it's probably always worth giving a go.
Thank you very much, both. Final radish question, perhaps. I don't know, some more might pop in. Dominique from somewhere in England is asking, is there a time limit from when you sow the seeds to when you lift the crops? She's noticed, for example, that radishes get bigger the longer they're left in the soil, but then they become a bit more inedible. So, Victoria, what are your thoughts on harvesting radishes?
Yeah, so you want to be harvesting the radishes every few weeks or so. We've actually designed the experiment so they can be sown in succession. And so long as you make sure you sow the same amount in your polyculture as your monoculture, that's fine for the experiment. Another tip is actually if you have left your radishes to go a bit woody, you can let them go to seed and then eat the seed pods, which are supposed to be really nice in salad and apparently less spicy than the root itself. So it might be better for those who are not keen on the hotness of radishes.
Okay, thanks, Victoria. A question here about growing experiments and when they start. Pavlos, Mike in Nottingham is asking via YouTube, is it important that all growing experiments start at the same time, or is that not so significant?
Thanks. Well, I think this depends on the type, again, on the type of experiment you want to do. Usually at citizen science projects, we want to engage lots of citizens into massive data collection. And for certain types of experiments, including experiments that have to do with growing and with soil and climatic conditions that change and so on, it makes sense to have this data coming at cohorts, coming at certain batches. That's why it is usually encouraged that a lot of participants join the experiments at the same time, and they share the data at the similar time slots, because this allows for comparability, but also for more data coming at the same time, which can be analyzed quickly and provide certain conclusions. Well, Grow is definitely a project that is keeping very good timing of its experiments, missions, and massive online courses, and we see a certain value in engaging large amounts of participants in doing the citizen science activities at the same time.
Okay, thank you, Pavlos. We've got another question on experiment timing from Laurie Heath, who's via YouTube. Victoria, Laurie's asking, I may not be able to do the experiment this year, but could definitely do it next year. Is that still useful to us? And can she register for the experiment now? Could you give us a little bit of information on that?
Yeah, so please do register as an observer, and then you can see how the experiment goes along. We are looking to do further experiments in future years, so it would be great if we could keep in touch, and if you could sign up to our newsletter, and we'll let you know all the other opportunities that come up. We're also planning some activities over the summer which people who are not doing the experiment might be able to take part in. So yes, please do keep in touch with the Grow team.
Okay, thanks, Victoria. Pavlos, we've got a specific question about timing in Greece. Maybe you'd be able to comment on this. Kiki Hattis, I'm probably not pronouncing that well, is asking us when the experiment will start. So in the UK, well, in Europe, it's starting this week, but what about in Greece? They're asking because they'd like to participate, but the time to plant these 3 plants in Greece has already passed. What's your suggestion on that, Pavlos?
So I think Kiki is one of the very first growers installing grow sensors in her vineyards, as I know. And I will be actually using her, be visiting her vineyard right after this webinar. So in fact, in this particular year, the growing season in Greece has moved a little bit earlier for almost a month. In the last couple of years, we have been observing really, really dry conditions with very scanty rainfall, at least in the northern part of Greece where you, Kiki, and me are located. However, these conditions are not the same for other countries in Europe, which are still a little bit colder and wetter. So the planting season is still due. However, this material that we are proposing, the protocols, the experiment protocols we are proposing, is not going to be on just for this year. Maybe next year we can plan it a little bit better and be able to plant those the radishes and beans and spinach a little bit earlier on.
Okay, thanks, Pavlos. We have a question here from Climate UK via YouTube about the list of flowering plants that you gave, Victoria. And they're asking if that list of flowering plants to encourage pollinators, they're saying it was very helpful, and they would like to have that available as a list. So can you comment on that, please?
Hey, so I'm glad you found that useful. I'll pop the list on the YouTube after the webinar. But actually, if you have a look online, there's some really good resources out there with lists of plants which are good for pollinators. The Royal Horticultural Society in the UK in particular have done quite a lot of research on which flowers attract the most pollinators and also different types of pollinators. So they're worth looking up. They've got a list there.
Okay, thanks, Victoria. And Alice, I know you've been doing some work and some study on pollinators and flowering plants. Have you got anything you'd like to add to that?
Hi, well, I think the key is to kind of, sometimes the best flowering plants for pollinators are what you could consider as weeds. So kind of maybe rethink weeds and your native species, and if you leave them to flower, they're really, most of them are really, really good for pollinators, especially because they grow all around in your local area. The pollinators that pollinate these species are already going to be there. So that's my advice.
Okay, that's great. Thanks, Alice. We've got a question here about spinach. Victoria, what are your thoughts on this one? Laurie Heath via YouTube is saying that spinach tends to bolt in our latitudes because of the longer summer days. In the experiment, should she just allow the spinach to bolt and measure its overall biomass?
Yeah, we've had quite a few people saying that spinach tends to bolt where they live. But one of the things we're hoping to find out are whether it actually grows better in a polyculture because of the shading from the beans. It might be less likely to bolt. In terms of measuring the yield, we really want you to measure things that you would normally be eating. So every time you take some of the spinach off to eat, we'd like you to weigh that rather than the biomass of the whole plant at the end. So yeah, don't include any bits which you're not going to eat, such as bolted bits. I hope that answers the question okay.
Okay, thank you very much. Just a general question, really. We are getting questions about how to encourage pollinators, aside from planting native plants and allowing our weeds to to flourish. Victoria, what other tips have you got for encouraging a bit more of those friendly invertebrates that we'd like to see in the gardens?
Well, definitely avoiding insecticides, because I mean, if you want to use it a little bit on some plants, it has an effect on any insect in your growing area. And I particularly like to try and leave some of my aphids on my plants, because I find if you leave them long enough, you'll get other insects like hoverfly larvae and ladybird larvae eating them, which helps out with your plant. But also, it's feeding those hoverfly larvae, which can, once they pupate into adults, will be helping to pollinate your plants. Yeah, so don't forget the larvae. That's my advice.
Okay, thank you very much. And we're also getting questions about mulching and about what mulches might be useful. So, for example, Jana Holmstead is asking about dry leaves from last year. Would they make a good mulch? Or should she use wood, twigs, grass cuttings? So could we have a few suggestions from you all about what you would use for mulching? Pavlos, you mentioned that you used grass cuttings. Could you give us a bit more detail on that?
Yeah, sure. So depending on rainfall during the winter, there are a lot of like wild grasses growing under the olive trees. For the last 10 years, a little bit more than 10 years, we haven't really digged the soils. And this is, so we constantly we are aiming to increase organic matter by cutting this understory vegetation after the flowering time. We want to attract the pollinators during spring, and then just before they start seeding, we remove them, we cut them, because we don't want them like to multiply in the next year. So we need to keep the balance there, because we also need to consider fire hazard as the summer days get hotter. So all this biomass that is left on the field during the summer is really, really beneficial, especially in non-irrigated crops like the olives, and in conditions of dry climates where there are water shortages and there is a need to start adopting more, let's say, water friendly farming practices. So mulch definitely keeps the soil cool while accumulating over time very, very important layer of organic matter. It increases soil carbon, it increases soil biodiversity. Like you can just go in a dry day and then you just like dig like the first 4 or 5 centimeters of mulch, and you already start seeing moist soil and even earthworms and other insects, all those that we call at organic farming the farmer's best friends. So mulch is working at all types of crops. We actually had a monthly theme last year on mulch, so there are a number of articles and stories and grower stories in our Grow Medium page. So just take a look on that and get inspired about how different people across Europe are using mulch to cover the soils and to enhance their life.
That's great. Thanks, Pavlos. We've got one last question that's come in, this time about companion planting from Inut Barbert, who's on YouTube, and is asking if we can tell them about using companion plants around the experiment plots in England, suggesting marigolds. Victoria, what do you suggest alongside marigolds?
Yes, so you can use any of your normal growing methods, including companion plants, in your experiment plots. The important thing is to remember to do the same thing in your polyculture and your monoculture. Otherwise, then it's really difficult and sometimes impossible to tease out what the differences are. So long as you use the same companion plants and the same amount in both plots, that shouldn't be a problem at all.
Brilliant. Thanks, Victoria. And thank you all for your questions that have been coming in. We've received your thanks for answering them. So thank you to you for asking them in the first place. And it's great to know that we've been able to help you out with some of those questions. Now we're going to move on to tell you a little bit about what's next for Grow. So I'm going to ask Pavlos to take us forward on that.
Thanks, Deborah. It has been wonderful to see all this interest in our online course and learn about people's stories and practices. It is really encouraging as we are moving forward with some very exciting opportunities to engage, learn, and grow together. We hope that many of our participants are joining us over the summer in the activities part of the experiment with polycultures and monocultures, and look forward to seeing them online in the Grow Forum and our monthly meetups. If you live in one of the Grow places, you can join in our Changing Climate mission. You can check that on our website, www.growobservatory.org. We also take this opportunity to invite you to join the last of our 4 free online courses this year, when we will revisit our Great Grow experiment. The course starts on the 5th of November on FutureLearn and looks at using and visualizing citizen-generated soil and growing data to make positive change happen in your growing space and in your community. This will offer a great addition to the things you learned in the last 3 courses. You are also welcome to register in our Grow Online Community Forum via the Grow website. This forum is all about bringing the community together to exchange knowledge on soil and good growing practices online. Your contribution also gives us the opportunity to learn from you, to help us design and deliver future themes and activities that suit you. For those who are taking part in the experiment, please make sure that you have signed up via the Grow website. You will receive emails with the links to the full instructions for planting, assessing your site, and measuring your harvest. And please do ask us any other questions you might have in the experiment section of the Grow Forum. And don't forget to follow and share our tweets, Facebook, and Instagram posts. All the links are on these slides. Most importantly, keep in touch with what's going on. Subscribe to and share our newsletter and bring your local community into your journey with Grow. Back to you, Deborah.
So finally from me, a big thank you to you all, to our presenters, our listeners, our participants, and of course our questioners. Grow is a citizens observatory, and that belongs to all of us, the citizens. And the more we engage, the more value we create for land, for science, and for our communities. So please do help us make Grow known to your friends and local community, practitioners, growers, local authorities, community gardeners, and whoever is interested in soil, food, citizen science, and environmental issues like climate change. You're all helping us build an incredible community of participants and partners, from government organisations to universities, research institutes, design and technology partners and NGOs, and of course you all. Thank you for your brilliant questions and queries. Do keep asking through our website and join us in our experiment this summer and our online courses this year and next. We look forward to working with you. Thank you. Till next time.